Welcome to Getting Started in CellEngine. I'm Anita, a technical applications specialist with the CellEngine team. This is the first in a series of videos that will teach you how to use CellEngine for cytometry analysis. Each video will take an experiment or a couple of experiments and go through part of the workflow step by step. At the same time, we'll talk about any tips or features that might help you as you do your own analysis. By the end of the video series, you'll be able to take CellEngine and analyze your own data, utilizing CellEngine's features to their maximum potential. In this video, we'll take a look at the layout and navigation of CellEngine, create a new experiment, and go over how to add information about the experiment as a whole. Then we'll upload some FCS files and add sample metadata, which is important for leveraging analysis in CellEngine later on. Let's go ahead and look at CellEngine. If you'd like to follow along with this video series, please feel free to go ahead and sign up for an account if you don't have one. There is a free 2-month trial available for anyone who is interested in trying out CellEngine. Once you have a user ID, go ahead and enter it along with your password and then click Login. Whenever you log into CellEngine, you'll see a screen that looks like this. There will be a folder hierarchy here on the left-hand side. Each folder can contain one or more experiments that you'll see listed in the middle of the screen. Each experiment is a collection of FCS files that will be analyzed together. CellEngine can handle large numbers of files. The largest experiment in CellEngine currently has over 30,000 FCS files. So we really encourage people to use files that share the same panel in the same experiment if you're at all interested in comparing the results. Before we jump to making a new experiment, there's a couple of features I'd like to point out. In the upper right-hand corner of your browser window, you'll see account options. Here you can change your password or email address if you ever need to do that. Next to account options is this question mark symbol. This will take you to two important places. The first is documentation. I'm going to right-click on documentation and open that in a new tab. Documentation will tell you about specific features in CellEngine and also includes some references for learning more. There are also a number of useful demonstrations and examples. These can be especially helpful if there's a particular figure that you'd like to make, but you're not sure how to approach making it. It's very possible that the steps will be right there for you to follow, along with an example of what the figure will look like when you're done. In addition to the documentation, you can also send feedback or request help using this question mark icon. When you fill out this form, it will be sent to the support team, including me and other members of the support group. You can submit feedback and ask a question. You can also send us a screenshot if you're in an experiment, or even share the entire experiment with us so we can see exactly what you're seeing. Please feel free to reach out to us at any time with suggestions or questions. Now let's make our first experiment. I've already got a folder set up for this particular video series. There are no experiments in it yet, so we'll create one by clicking New right above the main window. When I click New, it'll set up a new experiment and autofocus the cursor so I can immediately rename it to something else. I'm going to call this experiment Annotation Setup because most of what we're going to talk about during this video is annotations. Before we upload any files, let's review some other parts of the summary page. As you're setting up your experiment, you may want to add tags. Tags are searchable from the main page. If I click here or start typing, you can see what tags I've used for other experiments. I like to often add the cell type used in the experiment or the study ID. For this experiment, I'm going to add tutorial and PBMC tags so I can easily pull this up again later. The comments box allows you to enter any text freeform. Many users add comments to themselves about the analysis or comments to other collaborators working on the same experiment. Custom fields allows you to enter a pair of values. So for example, I could write study ID in one cell and then the ID value in the other. We'll talk more about revisions later on, but know that at any point during your analysis, you can click on Save Revision and create an immutable copy of the experiment, a snapshot of the experiment as it is now, which can never be changed. That can be helpful if you're planning on introducing major changes or if you want to track the development of analysis over time. As we scroll down the summary page, you'll see that the files list is currently empty, so let's upload some FCS files. Adding files to an experiment is as simple as dragging and dropping them into the browser window. I've got a folder here with some FCS files and a protocol, so I'm going to drop all of these into the browser window. You'll see these arrows in the upper right-hand corner have started spinning, and if I click on the arrows, they'll tell me what percentage of the upload is complete. You'll see the protocol and FCS files have begun to appear in the list of files down below. Anything that isn't an FCS file will appear here under attachments. This is a good place to add protocols, notes about the experiment, or any other files you want to associate with your experiment. While the files finish uploading, let me show you navigation and experiment sharing. I can navigate to other locations in the file path by clicking these links in the hotbar. So let's go back to the tutorial folder for a moment. Both folders and experiments can be shared with other CellEngine users. One of the big advantages of having a cloud-based platform is that it's easy to set up sharing. I'm going to share this entire tutorial folder by clicking the Share Folder button on the left bar. You'll see the list of other users that currently have access, along with their access level. I can change the access by clicking on this drop-down box and then hover over each of these options to get more information about what a user can do based on that role. If you have a custom domain set up, you can customize different user roles and create new ones. There are about 50 different permissions to different parts of analysis and different levels of modification. Those can be combined to create very specific roles for users if you have the custom domain add-on. One of the nice things about sharing folders is that experiments inherit permissions from the folder. So if I right-click on an experiment and choose Share, you'll see that the user I just added has access. This means you don't have to adjust the list of shared users every time you create a new experiment. You could potentially have everyone in a lab with access to all experiments by saving them all in the same folder, or set up a new folder for each study with the people who will be working on it. And then as new analyses get created, those will be shared. Let's go back to our experiment. You can see all of our FCS files have been uploaded now, so we're ready to move to the next step. You can navigate to different parts of the experiment by clicking these links in the sidebar on the left. If you ever need a little bit more space in the main window, you can collapse the navigation tab with these arrows at the bottom. That will give you a little bit more space to work. For the next step, let's go to Annotations, which is the tab where we can add information about samples. CellEngine organizes its analysis and file selection by metadata, so the more you add about each sample at this stage, the easier it will be to select different subsets of data and compare different groups in the next stages of analysis. If possible, doing that all at once will give you the most flexibility during the gating and figure creation process. There are several ways to add annotation information to an experiment. I can click and type in the cells. I know these cells have been treated with different cytokines, so I'll name this column treatment, and then I can type in the cytokines that I know have been used. You can also copy and paste from a spreadsheet that you have open on your desktop. If you have a larger number of files or you want to make sure everything matches without double checking manually, importing annotations will add information from a spreadsheet. Click on this cloud icon with the up arrow, and you'll see a dialog box. From here, you can upload or drag a spreadsheet into the window or paste values directly. I've got an example spreadsheet for this experiment, so I'm going to drag it into the window. It will show you the name of the sheet that it's importing. You can change these or adjust where the header is. Then click on Next. CellEngine will try to find columns in the spreadsheet and annotations that match. In this case, my example has the file names in it, but you could also upload information that is linked to other metadata. For example, you might want to add demographic information that is linked to a donor ID. So the source and destination column can be any column in your spreadsheet or experiment. Then choose the columns that you want to import. In this case, I'm only going to import dose and not time point. When you've made the selections, click on Import to finish. Many people include useful information about their samples in the FCS file name. If you click on this icon, you can add that metadata directly to annotations. This process works like text to columns in a spreadsheet. The file name will be split into parts by a character, in this case, the underscore. The files in this example have 3 parts. I'm going to uncheck Add Annotations for the first 2 columns because all these samples are PBMCs and from the same donor, and keep the last one, Treatment. I can rename the annotation column by clicking in the box. CellEngine will offer any columns that match, so I'll select treatment from the dropdown. If data already exists, I can choose to keep or overwrite existing values. Finally, I'll click on Create Annotations to finish the process. In addition to adding information from the file name, you can also add information from the FCS file header itself. This icon will bring up a list of all the data stored in the FCS file headers, which can then be added to annotations. This includes data like the cytometer used to collect data, the date of collection, start and end times, and other data. CellEngine has a validation tool to make sure that the information in annotations is complete and the values acceptable. Select an annotation column and then click on the check mark with the green dot to open the validation dialog. Here you can choose whether values are required for each sample. If the values are numeric, you can set a maximum or a minimum value, or you can add specific values. I can list exactly how I want treatment values listed, and then once I save the validator below, you'll see that it's highlighted any values that don't match. I can use these arrows to jump to any cells that need correction, and then click on the drop-down box to select from the list of acceptable values. This process makes sure that there's no conflicts when it comes to grouping files for analysis, and no files that are accidentally left out of analysis either. Now that we have some values for demonstration, let's talk about sorting and filtering values. You can click on the letter of a column to sort it. Clicking again reverses the order, and then clicking a third time returns you to the original order. Sort order depends on the values in the column. Generally, values will sort alphabetically or numerically, but CellEngine will also sort plate-well values by row or dates chronologically. You can change the sort order by clicking on these three horizontal lines. We'll talk more about changing the sort order when we discuss illustrations later on. For now, know that you can select one or more values and then use the move options to adjust the sort order manually. Annotations can also be used to filter files. When you see these three lines next to an annotation, you can use that to filter the values available. If I click on these, you'll see the list of values, and then you can check one or more to restrict the list. In addition to adding metadata, the annotations page has several other useful options. If I check change file names, then I can go to a file and change the name, which can be helpful if you're alphabetizing files. I can also concatenate files from this page by selecting multiple files with Control or Shift buttons on Windows or Command and Shift buttons on Mac. Then I can select the concatenation button, which will merge the files together. CellEngine will suggest a file name made up of common components from the source files. Since both of the files I selected had PBMC and unstim in the file name, you'll also see that in the suggestion for the concatenated file. When you're done, you can choose whether to delete the source files and whether to add a file number column to the output file. If you add the file number, a new channel will be created with an integer assigned that represents the file. Annotations and file headers will also be updated if the data is the same between files. You'll see here that common values were added to the dose and treatment annotations. So at this stage, we have uploaded files and added annotations from file names, headers, and spreadsheets, which concludes the initial setup of the experiment. In the next video, we'll move on to the gating tab, and there we'll go through the process of creating compensation matrices, adjusting channels, and basic gating. I'd like to thank you for watching. If you have any questions, feel free to reach out to us. We're always available via the question mark icon if you're in CellEngine, or you can contact us directly by emailing support@cellengine.com.